So what I will introduce to you today is a little bit more about crosses. So instead of just doing plain monohybrid crosses where we have one dominant and one recessive allele, I'm going to show you a couple of things that could happen or that you could be presented with that are a little bit different. The first thing that I want to show you is where Mendel's second law came from. It's the law of independent assortment. This picture can be found in a very, very similar looking state on page 593 of your textbook. Now what Mendel did was, of course, some monohybrid crosses, but he also performed some dihybrid crosses. Di means two. So a dihybrid cross means that we performed a cross based on two traits. In a monohybrid cross, we were only concerned with one trait. In a dihybrid cross, we're concerned with two. So here is an example of what Mendel would have done. Mendel took two plants that were homozygous for two traits and then crossed them. So in Mendel's cross, he took two homozygous plants, and this is for two traits, and crossed opposites. So in this example, which is the same one that's in your textbook, we'll show what happened when he crossed a green, tall plant with a yellow dwarf plant. So one of them was homozygous dominant for both traits. The other one was homozygous recessive. Now, just like in previous crosses, this is the P generation. Those are the parents. It's a pure cross because the parents are homozygous. The first generation, the F1 generation, just like in his previous crosses, had organisms that only showed one of the two traits. In this cross, everything in F1 were green and tall. So based on our operational definition of what dominant means, that means that green and tall are the dominant characteristic. So F1 were all this particular genetic combination. So everything in the F1 generation had one green gene and one yellow gene, because that's what they got from each of the parents, had one tall and one dwarf gene. But because they had at least one of those, that capital letter is what's making them appear green and appear tall, even though they still carry the allele for the recessive characteristic. Now in the F2 generation, F2 is this giant table down here. The F2 generation always gets made by taking two of the F1 organisms and crossing them. So to make F2, I took this as my cross. Two organisms from F1, I crossed them. This means that there were lots of possibilities for the gametes that those organisms could have produced. So I'm going to do a little bit of color coding here to help illustrate this. When we did a monohybrid cross, each organism could make two possible gametes, one or the other. Now that I'm doing a dihybrid cross, each organism can make four possible gametes because they have two characteristics and there's two alleles for each of them. Possibly this organism made a gamete that had the dominant gene for green and the dominant gene for tall. But it could have also made a gamete that had the dominant gene for green paired with the recessive gene for height. We could have also had a gamete that came from the recessive gene, so the yellow-colored one, but the dominant gene for height. And then finally, we could have had an organism that made a gamete that had the recessive gene for both characteristics. So because now we have two genes, and each gene has two alleles, 2 to the power of 2 means I have 4 possible gametes that I could make. Since I'm crossing two organisms that have the same genotype, I will have the same 4 possibilities down the side. Now, nothing is going to change about the way that you fill in the Punnett square. You're still going to look above, and you're still going to look to the side, see what letters you have. You're just going to have 4 letters total now. So here's what I'd like to highlight to you. The ratio. 9:3:3:1. That is the standard dihybrid cross ratio. That is the typical number that should show up most of the time if you do a dihybrid cross like Mendel did. Now here's what he saw. The 9 represents organisms that showed the dominant phenotype for both characteristics. So on this little chart, I'll just highlight them. Everything that's kind of on the outside and on the left has at least one copy of the dominant allele for both characteristics, as well as this guy and that one. So in total, if I count, there are 9 of the 16 possible combinations of offspring that would show the dominant phenotype. They don't all have the same genotype, but they would all have at least one copy of the dominant allele. The next 3, or the next number, the first 3, represents organisms that are dominant for one of the characteristics, but recessive for the other. So if I look, this organism, and this one all have the dominant phenotype for the first characteristic, but the recessive phenotype for the second. And then these 3 here are the opposite way around. So those 2 3s represent dominant phenotype for one and recessive phenotype for the other. So some plants will be green but short, other plants will be yellow but tall, so they'll have one of the dominant characteristics. Then Mendel noticed that there would be one. One of the 16 possible outcomes represented an organism that had the recessive phenotype for both characteristics. So based on this, Mendel's second law says that gametes form by independent assortment of alleles. If I were to put that in other words, he's saying just because you have the gene for tall doesn't mean you're going to have the gene for green as well. Each of those genes was inherited completely separately and randomly from the other. Now, I'm going to put a star here because although this is what he discovered, this is not always going to be the case. We will talk in future lessons at the end of this week probably about situations where it doesn't look like you can have any combination of gametes. We're saying it doesn't matter whether it was dominant or recessive, it's possible to get any combination of the parental gametes. Does anyone have any questions that they want to ask so far? So I have two other scenarios that are a little bit different from just a plain monohybrid cross that I'd like to show you. One of them is called incomplete dominance. And in this scenario, neither allele masks the other. So in my example, I have what's called a 4 o'clock flower, but this actually happens in quite a lot of flowers. The typical examples where this would happen usually have something to do with color or with size. In the 4 o'clock flower, you could start with two parents, one red one and one white one. And in the F1 generation, instead of getting all red or all white, which is what Mendel was seeing, only one characteristic would show up, he was getting all pink. It was like the two alleles mixed together. You didn't see all red, you didn't see all white, you saw a combination of both of them. So when we represent codominance, we generally represent the allele with a capital letter and then a little lowercase, like an exponent, kind of like what you would use in math. So the capital letter that you choose, I mean, really, I chose R here because I was looking at a red flower. I put a lowercase r for the red one and a lowercase w for the white one. We would assume that both parents were homozygous for their color at the beginning of this cross, so that it mimics what Mendel did. The result would be, in the F1 generation, that all four of these organisms would have one of each. One copy of the red allele, one copy of the white allele. And all the organisms have as their phenotype pink. If I were to do an F2 generation and cross two organisms from that generation, I would see a different looking ratio. So if you are doing a monohybrid cross and the ratio looks weird, you're getting numbers that you don't expect, that's a clue that it might be an incomplete dominance scenario. Now, like I said, usually examples about incomplete dominance have to do with colors. So you will need to remember what you learned in kindergarten. Blue plus yellow makes green, red plus yellow makes orange, red and blue makes purple. If you feel like you need a refresher on the color wheel, please let me know and we will practice. The other thing that I've seen fairly frequently is size. One parent is very large, the other parent is very small, the offspring are all medium-sized. That would be your other typical type of example of incomplete dominance. In humans, there's a couple of conditions. Sickle cell anemia is the one that gets used as the example quite a bit. In sickle cell anemia, there are two alleles. HB stands for hemoglobin. The A is normal, and the S is the sickle cell one. If a person had two copies of the normal allele, they would not have sickle cell. If they had one of each, they would have sickle cell condition, we would call it. It's not as bad. If they had two copies of the sickle allele, they would have the full-blown disease, sickle cell anemia, which is worse. So there would be varying degrees of the condition depending on if you had one or two copies of the sickle cell gene. There's another condition called hypercholesterolemia, which is another medical condition where if you have two of the gene, it's worse. If you have one, it's not as bad. And if you have none, you don't have the condition. The other example, or the other scenario that I'd like to mention is codominance. In codominance, both alleles are fully expressed. So that means one does not mask the other, but they also do not mix together and make some intermediate color. In codominance, you see both alleles. So this is a picture of a roan horse. And a roan horse, if you were to look very closely at their, what do we call this, fur, hair? What do you call it on a horse? Fur? hair, coat, their coat. Let's look at their coat. The stuff that's covering their body, whatever we're going to call that. It looks like maybe from a distance they might all be one color, but if you look closely, there are actually two different colors of hairs. Some of them are a kind of browny color, and some of them are a kind of white color. They are not kind of brown, kind of white. They have some fully white hairs and some fully brown hairs. So both alleles get fully expressed. In other words, you see both colors, not a mixture. In codominance, the best example I could give you in humans would be blood type. If you are blood type AB, it means that you have the allele for blood type A and the allele for blood type B. So your alleles look like this. A doesn't cover up B, B doesn't cover up A, and they don't mix together to make some other random letter that I guess would look sort of like I don't know what letter that would look like, a messed up letter. You see both of them distinctively.